Guys hear okay?
I can hear you perfectly. Thank you.
Great stuff. So thanks for having me. I really appreciate it. Let me start sharing my slideshow here, and then we can get cracking. All right. It's thinking.
Awesome. We can at least see it, which is a good sign.
Okay, can you guys see it?
Yeah.
Okay.
It's just not full screen, but we can see the slides.
That's odd. Let me just hold on because it's not. There we go. There we go. How's that?
Wonderful. Thank you so much.
Great.
Over to you.
So as Alana said, I'm in the Department of Conservation Ecology and Entomology with Stellenbosch University. I'm not an entomologist by training, I'm a geneticist, but I found myself falling into the Polyphagous Shot Hole Borer study scene very serendipitously. There was some funding available for this project, and I'm going to start by telling you a little bit more about what I do in the landscape genomics concept. And then I'll expand a little bit into the bigger scheme of shot hole borer research that's being done to give you an idea of how much work is going on at the moment, and to answer a little bit of the very common questions that people often have around this beetle. So the big title of my talk, a Coleopteran Conundrum. So Coleoptera is the family of beetles, and this is quite a conundrum. So it all started with the observation as part of a Sentinel project here in Pietermaritzburg. Sentinel projects deal with using botanical gardens to monitor tree health through whether that be introduction of invasive species or new diseases cropping up. But botanical gardens serve as great areas to monitor to see how nature is doing health-wise. And that's where these little beetle borers were noticed for the first time. And here you can see a tree that is very, very heavily infested, little shot holes pickling the tree, if you will. And that's where the common name comes from, shot hole borer. Its Latin name is Uwelaceae fornicatus. So these shot holes are very, very small in size, only about 1 to 2 millimeters in diameter, and they're quite characteristic. So there are a lot of beetle borers that are both native and some other invasive ones, and people often get confused when they see just random holes on trees. They think it's shot hole borer, but you've got to be careful, and it's wise to take a little bit of a measuring tape or something. They should be around 2 millimeter or about 1 millimeter in diameter, and they are perfectly round most of the time, as you can see over here. So here you can see an oak tree that was felled up in Santon, more than 50 years old this oak tree was. And you can see the little, it's almost like little squiggly lines that's going in from the outside towards the heartwood of the tree. And that's the galleries that these beetles make as they enter the tree. They go through the bark in through the sapwood, but they'll never enter the heartwood because that central heartwood is obviously not it's quite hard and it's quite dead. So they often just found in the sapwood on this outside diameter, as you can see in this image. So here's our little culprit. This is the shot hole borer. This is actually the first one I managed to sample myself next to a ruler here. Just tried to take it with this cell phone. Microscopes take much better pictures. And as you can see, they are really, really tiny little critters. So the female beetle is the larger of the two, of course, and she's about 2 millimeters long. And you can see the detail here, like little hairs on them to as they burrow through trees to get the sawdust and things off of them. The males are quite a bit smaller, almost half the size. And that's very characteristic. They're always smaller. The males are also much less abundant than the females. Males only occur about one for every 20 females, there's one male. So the reason for this is their breeding strategy. The shot hole borer is what we call haplodiploid. So this means that females can enter a new host tree, and without mating, they can produce male offspring. And these male offspring have just got one set of chromosomes, but they're functional. And with the male offspring, then they can mate back to that female that entered the tree, producing more females to mate with or to produce males. And the females then have two sets of chromosomes. So very interesting breeding structure and very clever, because this means that females can disperse between host trees without having to worry about dragging a male along. In addition, this is because males don't disperse. Males tend to stay with the tree. They might climb out of their little hole and scramble up to another hole and go in there, but they cannot fly. The females, on the other hand, they can fly, and preliminary studies have shown that they can travel as far as 2 kilometers in one go. But this is very old research, and we are actually currently underway investigating a little bit more around their dispersal, but I'll talk about that later. So these little beetles bore into the tree, and one of the signals is signs, aside from these little characteristic shot holes that you will often see, is what we call frass noodles. So in this image, you can see it's almost like a sawdust-like structure that is found all around the entry holes, and it makes these noodle-like shapes. And that's because as the beetle bores into the tree, it pushes the sawdust that it makes through boring, pushes that out. So it doesn't eat it, pushes that out, and it makes these frass noodles. When you see frass noodles, you know that the infestation is current. It's probably going on as you're watching, and it's quite fresh. So you know that that infestation is happening in that moment. It's not an old infestation. The beetles then also make these galleries, as I showed earlier on that oak. So what they do is they now bore through the bark into the tree. You can see these little tunnels that they've now made into the wood. And these galleries are where they then go and lay their eggs and rear their offspring. And they will have often multiple generations within a tree before they decide that they've now had enough, and the females will climb out again of the tunnels and disperse to a neighboring tree. So they can often stay in the same tree host for many, many generations. And a shot hole borer generation lasts about 40 days if the temperatures are optimum. If under colder temperatures, that cycle is longer. And under warmer temperatures, that cycle can often be a little bit faster, but 40 days is the average. So the interesting thing about the Polyphagous shot hole borer is this discoloration that you find around its little tunnels. And you'll often see that scientists, in order to diagnose whether it's actually shot hole borer, they'll chisel around the hole. So this little picture I've got here, you can see the bark has been chiseled away. We've got our hole there in the middle, and there's this little black disc of discoloration around the hole. That's caused by their fungal symbiont. So the fungal symbiont is called Fusarium eualaceae, or just Fusarium for short. And this fungus they carry with them wherever they go. They have a special, I think you could call it mandibular structures, the mycangia, which they keep these fungi inside, disperse to a new tree, and literally plant new fungus in these little galleries that they've excavated. So the magic about this little beetle is it doesn't actually feed off the tree. It feeds on fungus. And it's actually one of the very few insects that do fungi culture. Most people know the examples of the ants that provide leaf litter to grow fungus, and they actually feed off this fungus and they cultivate it. Now this ambrosia, this ambrosia beetle, the Polyphagous shot hole borer, does exactly that. It grows the fungus inside its tunnels where it rears its larvae, and the larvae and the beetles feed off of the fungus. So often the beetles will only decide to disperse to a new host tree when the tree host can no longer support their fungal symbiont, because without that, they have no food and they move on. So of all the signs of the shot hole borer, the most characteristic sign that that's what you're dealing with is this blackening around the entry holes, which is caused by the fungus. So for my study, we firstly wanted to know, well, where do these guys come from? And now that they're here, where do they go? And in this image here on the right, quite a nice one, you can see it's a branch of Albizia, a flat crown here in KZN. And you can see the galleries that they've made, and you can see it's just jam-packed with little black beetles all stacked in there. And that's them having burrowed in, made their galleries where they now rear their young. So for my study, we collected firstly samples, individual beetles, and here you can see them all stored in ethanol. And we collected beetles from across South Africa. We wanted to cover pretty much every single province, and we wanted to collect from a range of host trees. So let's pause there quickly and just discuss the host trees. You'll often hear that they talk about reproductive host trees and non-reproductive host trees. So reproductive host trees imply that this is a tree where the beetles will bore into and they will stay for a while. They'll produce offspring, they'll hang around, they'll eat their fungus, and within time they will disperse onto another host tree. And those trees very often take quite a hard knock, and they often die. Non-reproductive host trees are trees that we have observed shot hole borers entering. We've seen they make tunnels. We've seen a little bit of gallery formation, but we have no evidence that they're actually breeding in there. So they seem to go in, try it out. Perhaps their fungus won't grow. Perhaps they don't like perhaps the tree has some defenses they don't like, and they eventually leave. And these trees, sometimes if the fungus doesn't establish, these trees will often survive quite fine. So there's this distinction between reproductive hosts and non-reproductive hosts. So we were indiscriminate. We sampled borers from pretty much any tree we could get our hands on, physically removing them from branches. So we could associate what beetles we got with what trees. And here you can see a little bit of a map. The size of the whole circle represents how many beetles I obtained. And you can see KZN is very well represented because that's really the starting point for this beetle. And we have one little dot there in the Northern Cape, Young Kempdorp from the pecan nut orchards. And then each slice of these pie charts are the different host trees. So you can see in KZN, that big slice of green was Afzelia, and also quite a few Indigofera that I sampled from. And this will often be either a tree with an abundance of beetles or a copse of trees that you sample from. Whereas in the Western Cape down there, you can see Acer nugunda, the top purple. That Acer nugunda is very heavily infested in the Western Cape, and we got quite a lot of samples from there. And then, of course, other popular tree species as the poplar trees, Populus, and of course, oak, Quercus robur. Right, so we had this sampling strategy, but we went one further, and we also collected, well, not collected, we obtained beetle genetic information from other countries, because we do know that Eualaceae fornicatus, the species, is native to the Asias. But there are quite a few of them there, and they are available on genetic public databases. So we went and obtained some samples off of these databases that with known locations, so that we could add them to our data set. And this included places like China, Vietnam, Taiwan, Japan, and also a few outliers there in the US. So you'll see in California, there's a little dot. So California has the same shot hole borer infestation that we do. And then in Madison-Dade County and San Diego, there are two other borer beetles that we just used to sort of as an outgroup. Of course, then we did some sequencing. So for species identification in these beetles, you can sequence a small section of DNA called cytochrome oxidase. And here you can see little color lines depicting where there are differences. And these point differences are enough to tell us, is it the same species or not? Because the same species or haplotype, I'll explain that in a bit, they will have identically the same sequence. If there's a difference, even if it's only 2 or 3 bases that differ, then we're looking potentially at something else. So you can see here a little collection of all our samples and where there are some sites of difference. So after collecting hundreds of specimens, cataloging them, photographing them, knowing which host trees they come from, and confirming their species identity with cytochrome oxidase, we drew up a genetic tree. So these genetic trees can look a little bit daunting. So I've got lovely purple arrows to highlight the important bits. This first arrow that I'm showing to you, this is what we call haplotype 33. So a haplotype is within the same species, a slight genetic variant. It's often associated with the mitochondria often. So some of you might have heard of, there are a few, 7 human haplotypes. So it's seven mitochondrial groups that people can be grouped by. So similar here, there are a number of mitochondrial groups that these borers can be grouped by. And this number H33, this is the one that we have here in South Africa. This is the one that was discovered way back in 2017, 2018 in Pietermaritzburg's Botanical Gardens. And it is also the same one that is present in California. And this little guy comes from Vietnam. So there's haplotype 33 is confirmed to be in Vietnam, the US, and then here in South Africa. That's a little bit what everyone knew and what everyone expected. However, we found a second haplotype, which we're calling H38. So H38 is also seen in China and Vietnam. It's difficult to know which one of those two countries is its true native range at this moment, but the fact remains that we have a second Polyphagous Shot Hole Borer haplotype in South Africa. Okay, so that's a lot of detail and haplotypes and the different species and things. What does that really mean? That means that we are looking at a changing genomic landscape in South Africa. That means that we probably didn't just have a single introduction of this invasive species from one location. We probably have had multiple introductions from multiple locations. And that already alludes to a little bit of a problem in that these entries of invasive beetles into the country are not rare. They're probably pretty common, and that it takes us a long time to pick up on them. So the first time Polyphagous Shot Hole Borer was observed was in 2017, 2018, but it's probably been here since 2012. And similarly with this new haplotype we found in my study, it's still only present in KwaZulu-Natal, whereas the other one is spread throughout the whole of South Africa. But who's to say that it will not mirror the original haplotype and also spread throughout the whole country? In addition, we're not sure how long it's been here or what the difference is between the two. So morphologically, they're very similar. Genetically, they're quite similar, slight differences. But is there difference in host preference or in virulence to trees? Those kinds of questions now definitely need answering. So this little study of mine has been a pilot to a larger study. So the little sequence I showed you is a small pinprick on the bigger genomic map of these beetles. And what we've done at the moment is we've done what's called a whole genome Snip analysis. That means we've taken its entire genome and we've looked at every variation in it to draw up a very, very detailed map of the differences between these beetles. So that data is currently being analyzed. This is the last year of my postdoc, so we're hoping to have some results at the end of this year. And with that study, we'll be able to answer some questions as to, is there perhaps some kind of host preference, native versus non-native host trees? Is there perhaps some kind of difference in virulence, or are they actually really quite similar? We might also be able to track whether this is truly a second introduction or has it been an in-country mutation? Those kinds of questions we're hoping to answer. So I'm hoping that this gave you a little bit of an idea of what the power of genetics can do at the moment in terms of shot hole borer research, and a little bit of background on them, of course. So now, just to touch base on what the bigger group or the bigger community of research is doing. So we call ourselves the Polyphagous Shot Hole Borer Research Network. We are based the headquarters, if you will, are based at FABI, which is at the University of Pretoria, and encompasses a number of universities. Almost all universities are participating in some way at the moment. So here's just a little pie chart to show you what kind of projects are taking up a big chunk of precedence at the moment. You'll see we've got monitoring, and these are very important, just literally boots on the ground, doing field work to see how the host tree species are changing, where new infestations are taking place, what the infestations look like. Then we also have quite a bit of work happening in biological control, which we'll touch on just now. The general biology, my students personally are working on some of those questions. How far do these beetles disperse? What's the effect of temperature on their dispersal? And that ties in very nicely to climate change. And then, of course, how far, as part of the dispersal, how far can they actually fly? We've got studies on agricultural crops, pecan nuts, avocados. All of those guys are very worried about the effects of the borers. Urban and as well as natural forests. We all know the devastation that's been caused in Johannesburg urban forest. And then the question always arises, well, how long before these beetles become a real problem in our native trees, which they're really just touching on at the moment. Can you guys still hear me? It's fine.
Yes, perfectly. Okay. Sorry, I think someone came unmuted, so I've just
that's fine. Again. All right. Yeah, so that's just a breakdown of what the larger scale research group is doing. So you can see we're really trying to tackle this problem from every single angle, and have some really good teams working on these questions. So to give you some data from our research group at Stellenbosch University, there are currently 10,000 trees in Somerset West that are highly susceptible. And when I say highly susceptible, I'm talking, you know, the oaks and the Acer nugundas, where once these beetles infest, it's 2 summer seasons before the tree is so diseased that it will almost definitely not recover. And these trees have to be felled, they have to be chipped, and solarized or burnt. In Stellenbosch, it gets a bit worse. There's about 20,000 trees in Stellenbosch that are highly susceptible. We all know Stellenbosch has a lot of oak trees, a lot of plane trees, and that's quite a problem. The beetles have definitely moved over into Stellenbosch recently, and we have had a couple of sightings confirmed. There's one of the studies of a PhD student in our team is looking at injecting with poisons, and this will be combinations of fungicides and insecticides and or chemicals that will boost tree health. So these chemicals have found sometimes to curb infestation a little bit. And again, it's in some tree species and not in all. So there's really no silver bullet that this study has found as of yet. The little graph here at the bottom, just to show you, you can see data all the way from 2019 to currently. So this is on Vergelegen wine farm in Stellenbosch, which has a lot of older, not native trees, but older non-native trees, which are brilliant hosts for the beetle. And you can really see the numbers just skyrocketing and unfortunately not abating just yet. The last count from 50 different traps had a count of 5,000 beetles that month, which is significant. What you'll notice as well is that the last two, or from about February 2021, the data seems to make 2 very obvious peaks, and this just corresponds to the summer months. Sampling these beetles is very obviously starts in February when it's starting to get really nice and hot. Peaks around about end of summer season, and then June, July, August is usually very quiet as it's too cold and the beetles don't really move around very much. So then in terms of control, that's always the question that most people ask, oh, but this company is providing some kind of benefit, or that one says they have a poison that works. And the short answer is that there's no chemical product currently registered, that was to say legal to use, on Polyphagous Shot Hole Borer in South Africa. So anything that is available is not registered for use. Light infestations are definitely achieved, controlled by direct injection with insecticides and fungicides, because remember, this is a twofold problem, because you have the beetles boring into the tree, so they do cause some damage. The tree eventually struggles to transfer water because the beetles have bored so much, but you also have the beetles planting the fungus in the tree, and it's really the fungus that kills the tree. So you have to tackle it with a combination of insecticide and fungicide. So direct injection into the holes will often be a benefit, but most large organizations find this, or even homeowners find this quite expensive and quite tedious. There are some combinations of these insecticides and fungicides that have proven beneficial and are being investigated. And then there are a number of studies at FABI also currently looking at more biological control options. Because this is an invasive species, we're really not sure what predators are supposed to be tackling it. So we're investigating things like parasitic wasps, looking at the mite contingency that these beetles carries, just to start understanding in terms of their biology, is there anything a little bit more natural that we could use to get the numbers down? As a side note, if we look at the infestation progression in California, where California is about 2 years ahead of us in terms of the infestation. And what happened there is they had very, very bad infestations. And after 2 years, 3 years, it settled down to a little bit of a homeostasis where the beetles aren't eradicated. All of the weakest trees have died. And now there's low level, semi-manageable infestation in their summer months. So we are predicting that it could go something along those lines. But as I said, that's California. You can only predict. You can't always say that that's 100% guaranteed. So that's basically my work, the Greater Research Network's work in a nutshell. And I thank you for your time. If you want any more information, do go look at this website, Fabinet /PSHB. They have a lot of info on how to tell if your tree is infested, who to contact, interesting articles, as well as a list of the current research being done. Or you are more than welcome to email myself. I love answering questions. And then, of course, this work is funded by the CIB. So thank you very much for your time.
Sure. Thank you so much, Anandi. That was fantastic. I really enjoyed it. You did such a brilliant job, and your research is so interesting. Yeah. So thank you on behalf of all of us at Friends of Tokai Park. That was amazing. I see that we've got at least 4 comments in the chat, and I think that most of these are questions. So I think, if you don't mind, I'm going to ask you those questions one by one. And
yeah, sure, go for it.
And for the rest, if you'd also like to ask your questions, please do pop them in the chat. I mean, this is the perfect opportunity to get an expert to answer all those questions you've just been dying to have answers for. I think maybe also what I'll do, I will ask each person if they'd like to unmute and ask their question themselves. And if you don't want to, I'll just read it for you. So I think, Becca, you are first. Would you like to unmute and ask your question directly?
Of course.
Okay, great.
Good afternoon, good evening, guys.
Hi.
Good evening. Hi, Ananda. I'm sorry if I pronounce your name.
That's okay.
Yeah, my name is Bierman, and I actually got a chance I think last year there was people from FABI that came to DUT, and they did an introduction about Shothole Borer, and they took us to some of the sites in La Lucia, where they've been examining it and are studying it further. So the question really is, does this Shothole borer have like specific trees? Like, do they have preferences, or do they just go about which is the shortest tree, or do they like have some type?
They very definitely have preferences. So you'll remember I mentioned the reproductive versus non-reproductive hosts. So they'll often go for those reproductive hosts first. That's the stuff that works for their life cycle. It grows the fungus really well. And this is stuff like the poplar trees, the box elder, thank you for the English name, the oaks, those kinds of trees. That's their first preference. But what we have found, there's Willem de Beer did a very good little study in Jan Kempdorp, where there was a host of oak trees, which obviously the beetles were breeding in. And then they would fly out from the oaks into the pecan-nut orchards, and they'd sort of look around and come back. So they weren't breeding in the pecan-nuts, they were just infesting a little bit and coming back again. They then removed those oak trees, which were now the preferential host. And what they found is that the beetles are starting to breed in the pecan-nuts. So they have their preferred hosts, very definitely, but we're not sure yet what they will do if we put them under pressure. How flexible are they in their tastes, basically?
Thank you so much. It seems really important to find biocontrols. But I was thinking, like, are you looking for something that is native, or are you willing to expand exotic?
No, so I think the most of the focus at the moment is native. But it's in its early stages. So I must say they've only started with some of the parasitic wasps and such. They really haven't gone through a long list of candidates yet. So it's early days.
Yeah, the biocontrol is very interesting question. Yeah, Becca, 2 great questions. Thank you. So now I've got a question here from Nkululeko. Would you like to unmute and ask your question? Or would you like me to read it? Just trying to give a chance. Okay, I don't see anything. I'm just struggling to find in the participants list, but I'll read the question then. So she says, "Hi Anandi, your talk is brilliant, thanks. I've seen those big plane trees in the avenue of the Pietermaritzburg garden and the amount of infection.
Yeah.
Do you know the rates of infection of those trees? And is the infection fatal for those trees? And if so, for how long are they expected to survive and stay alive?"
So those trees actually, I'm glad you've gotten to see them. They're really impressive. They're some of my, I'm not a fan of non-natives, but those are my favorite. Those trees are a very interesting example because that's actually where the Shothole borer was discovered the first time in 2017-18. And you'll see those trees are still around. They're actually still looking pretty good. And what happened there is the Shothole borer entry holes have healed up and they make this thickened callus over. So those trees are really bumpy now in comparison to a lot of other planes. And I've been there every year since 2020, and I've watched the infestation go from the bumps plus a few new holes to no new holes and just the bumps. So very clearly, the beetles have infested those trees, moved on, and there's no new infestation, and the trees are surviving. So that could be any number of things. That could be the trees are just so big, there wasn't a high enough infestation level to really do them damage. It could be that the environment there is just so perfect, they can resist that infestation and just carry on living. Because we do see plane trees in other areas that do die. So those trees are a very nice case study of the length of infestation from its start to its finish. So in that instance, you know, in the span of 2018 to now, 4 years, the infestation has come and gone and the trees are surviving. But that's definitely not the case everywhere, and it's definitely not the case with every tree species.
Yeah, great. Thank you for that answer. Very interesting. I didn't know that trees could survive the infestation.
Yeah, they do sometimes. Erythrina is a classic example. They're so tough. They simply wean off the infested branch and they carry on like it's no problem.
Amazing. Thank you, Anandi. We've got another question here from our chair. You looked at the genomics of the beetle, but what about the fungus? Is anything known about the origins and the types of fungus? And is it perhaps, is it not more than one fungus, perhaps?
Yeah, that's a great question. So yes, of course, we're looking at the fungus. So I'm more a beetle girl myself, but colleagues of mine at FABI are looking more at the fungal side. So there they will actually, like I'm sampling beetles, they will scrape the bark and sample fungus. And they find that the shot hole borer here is carrying one symbiont, that's Fusarium, but you'll see in literature that there are two other symbiotic fungi mentioned, and other Uwalaceae beetles do carry more than one fungus, and we're not even always sure what all of them do. So one of the fungal species, we're not quite sure, it's not being eaten, it's not being farmed, but it is found associated with the beetle. So there's definitely some other benefits there. So they're investigating that side. We're also looking at fungal exchange. So now that we've got more than one invasive in the country, do they carry different fungi, or can they swap them around? Those are the kinds of questions we're looking at. And then I myself have, in addition to the genomic landscaping, we've sequenced the genome of the beetle, and we're also doing transcriptomics on the fungus. And that means we're looking at the communication between the fungus and the beetle at a whole genomic level. So it's early days on that work, but the fungus is definitely not a player to be ignored.
Interesting. Sorry, Chair, I didn't give you a chance to ask the question yourself. Did you want to ask anything to follow up, or are you happy? Not unmuting now. Oh, there you go. Happy. Okay, good. Didn't want to steal your thunder there. Karen, would you like to ask your question yourself, or would you like me to read it? Karen Sternberg.
Please, could you read it, Alana? Thank you.
Yeah, sure thing. Okay. All right. So Karen asks, I was under the impression that the beetle feeds on the cambium layer of dead or dying trees and cannot kill a healthy tree. So, for example, after many years of drought, so many trees were severely stressed. Then the rains came, and the tree roots, which had cracked from dryness, now sit in water. Soil-borne fungi then enter and kill the tree, and the beetles colonize it. Eventually, the beetle will run out of food and just tick over. Could that not be the case?
That's a very interesting way of looking at it, because it really ticks all of the boxes that we've seen. I mean, Johannesburg is a classic example. Those trees are in an urban environment. They have had a stint of drought, same as in the Cape, so they are stressed. We get the rains. So that does make sense. I think where the connection really comes in is finding the association with these things. So you see the beetle entry holes. So as you say, that could just be happenstance. The beetle is just entering. But then we have also isolated fungus from the beetle's mycangia. So we very definitely know that they carry this fungus. We isolated that very same fungus from scrapings on the inside of galleries. So you can see it, it's a black fuzz on the inside of the galleries. And we could match what we found in the beetle cheeks, if you will, mycangia. We could match that to what we find inside of the galleries. And we've also, we can rear these guys in artificial conditions, and we know that they eat the fungus. So if they were eating cambium, our artificial rearing system wouldn't have worked, but they very definitely eat the fungus that they grow in our artificial scenarios. So that shows you how tricky it is to puzzle these things out, because it's all of these tiny little building blocks that lead you to make a conclusion. So that's just the train of thought that helped us understand how this works. But this shot hole borer is not the only beetle. We call them Ambrosia beetles as a group, and it's very well established that this is their modus operandi, that they infest trees, they feed on the fungus. the question, as you then say, is it then the fungus even that's killing the tree? I am not the fungus expert, so I know Fusarium dieback is the very typical dying of the branches at the top, but I actually can't give you what the link is between us understanding that dieback is caused by the fungus. So that's one hole in my knowledge base, I'll admit.
Very interesting. Thanks. Oh, go ahead, Karen.
Thanks very much, Ananda. I was just wondering, does the fungus not feed on nutrients like sugars, and then the beetle feeds on the fungus? It doesn't actually feed on the cambium.
Yes.
So the tree is actually dying anyway, and the shot hole brings in its fungus, the fungus feeds on the nutrients, the shot hole feeds on the fungus, but doesn't actually cause the death of the tree.
So yeah, so it's not the beetles themselves that are, sometimes the infestation is so heavy that that poor tree looks like Swiss cheese, and it's obviously cannot even transport water up the phloem and the xylem tubes because of so many galleries. And in that instance, those trees, you know, it's a physical damage kind of thing. But when you have a mild infestation, yes, it's not the beetle that's killing the tree.
So just on that note, obviously it would be so much better if we didn't have to use poisons or toxins on trees or in our environment, because it affects so many other species.
Yes.
So perhaps if one could look at that aspect and avoid the chemicals altogether would be great.
I must admit, I give the information about the chemicals because that's honestly, that's the first question we get. But what can we spray? And I'm not an advocate of spraying things. It's also bad for us, you know. So I'm putting a lot of money on the biological control or on nature running its course, but you know, we can't always just leave things. So it's very tricky. And luckily, most of the time, these chemical solutions are too expensive for people to apply.
Yeah, too expensive also for our health. But thanks very much, Anand.
Sure, you're welcome.
Thank you, Alana.
Yeah, thanks from our bee experts. So it's nice to hear these questions. Maybe it's worthwhile chatting further. So yeah, thank you so much for that interesting question. So Ian Preston just says, thank you. That was a fascinating talk, Anandi. And then Tony Rebello says the cambium feeders are different species of borers, probably longhorn beetles. I don't know if you want to comment on that.
So exciting. We have a project running at the moment funded by the NRF. It's a foundational biodiversity study. And we're expanding with all the sampling that I did in KZN. To give you an idea, I collected, before I knew better, I collected 400 different beetles from wood. This is by, you know, removing bark or chipping away at pieces or whatever. And of those 400, only 200 were actually Polyphagous Shot Hole Borer. So that combined with the fact that we know very, very little about wood boring beetles in South Africa. We've launched a pilot study here in KZN. It actually kicked off this Wednesday. And we are going to investigate native wood boring beetles to get a better understanding of what's out there and where they are.
That's very cool. Yeah. And maybe, yeah, just worthwhile actually mentioning. So we've talked about Stellenbosch, but Anandi's sitting in KwaZulu-Natal at the moment, giving this presentation. I don't know if you were there for field work or.
Yes, I was actually out in the field all day today.
Okay, very cool. We've got a question here by Ian. Ian, you're very active on the chat, so would you like to unmute and ask your question and interact a bit with Anandi?
Sure, thank you. I just wanted to know, have any of these beetles been discovered on the Cape Peninsula? And then Tony said yes, but not confirmed. I see later in the chat.
So yeah, excuse my ignorance, whereabouts or what area ranges the Cape Peninsula? Because I work mostly in Somerset West and Stellenbosch. That's our main focus at the moment.
Across the False Bay, other side of False Bay, with Table Mountain down to Cape Point.
Actually, not really. So we have traps and a monitoring system out Kirstenbosch way. And there's not been anything as yet.
Okay, thank you.
Yeah.
Great. I want to come back to Sally's question, if I may, Sally, so you can maybe prepare to ask your question in a minute. Just because right after that, the conversation continues in the chat where Tony Rebello shares a link to say that there are some records of the Polyphagous Shot Hole Borer, but they're not yet confirmed. So that might be interesting. I don't know if someone can go out and check it. Tokai and Sweet Valley, and then there's a link there. A website EDRR. I'm not sure if you know about that and how, whether that's a reliable platform. And
yeah, I'll have a look at that link just now. But basically, when it says not confirmed, Fabi runs a service where you can send them either the sliced wood samples where they can isolate the fungus from, or if you're lucky and you find the beetles, you can send them that. And they will do this PCR of the short genetic segment that I mentioned tonight to confirm whether it's shot hole borer or no.
Okay, that's very useful. Thanks. Tony, do you want to unmute and add anything on that before we go over to Sally? Okay, perhaps not. But yeah, that'll be great. Oh, there you go. Okay. Anandi, feel free to check out that link later, and then hopefully those records have indeed been submitted, because it will be interesting to know. Okay, Sally, over to you. Would you like to ask your question about burning of infected?
Yes. Can you hear me?
Yeah,
perfectly.
Yes. Anandi, first of all, from a non-scientist, thank you for a talk. I mean, that I could understand, you know, with you being a scientist. I'm really grateful for that.
You're welcome.
My first real awareness of this was 2 years ago when we went to the Marlets Reserve near Swellendam. And one of the first questions they asked us when we were checking in was, had we brought any wood for the braai? Yeah. Because if we had, they were going to confiscate it. And we said, well, no, we actually don't travel with wood because we know about these borers and we've only got charcoal. And so it just made us aware that burning this wood is not a way to get rid of the infestation. And I just thought maybe if, you know, that hasn't been mentioned, maybe it's just something that is quite an interesting point.
That's actually a good point you raised there. So the wood burning or the wood transporting is just one of the mechanisms we suspect that these beetles have traveled so far. Because think about it, from Pietermaritzburg, well, no, from a harbor, either Richards Bay or Durban, we're not 100% sure. So from a harbor to Pietermaritzburg Botanical Gardens, all the way to Jan Kempdorp, how on earth did this little guy make it so far? You know, 2 kilometers of flying every now and again is not going to cut it. So we definitely suspect that they travel with people, and the most obvious mechanism is in firewood, because they will burrow into something, even if it's dead or if it's not a host, if they're seeking shelter or whatever the case is. So the taking wood up and down has been one of the sort of red flags that a lot of people have adopted. And what happens is, if it's still a good sized log, they're burrowed so far in that by the time they feel the heat, they just escape and they fly off. And once they fly off, they can disperse to any other tree in that area. So taking bigger logs and burning them is not a solution to getting rid of infested wood. What the recommended method is at the moment is to chipper it about 2 centimeter blocks, which is a pain because that's quite small. Very few people have chippers that can do that. But you chipper them and you have to solarize it, which means you put that chipped wood under a black top hole and in the sun, and that bakes the majority of them. And then that chipped wood is then safe to use either by burning that or any other way you want to get rid of that.
Thanks very much. I mean, it's just a warning, I think, to all of us. You know, just use charcoal, you know, because if you're going somewhere, because, you know, it's so dangerous to carry the wood. Thank you for that explanation. That was great.
Sure.
Yeah, thank you very much, Sally. And then we've just had a few other people saying thank you. And and then a few comments again by the chair. So I'm going to give the chair the floor at the end to thank you. So maybe he can add a few of his comments there. Are there any other questions from the participants? Luyanda, you've raised your hand. Please go ahead.
Hi, Anandi.
Hi.
Thank you for this talk. I also did a research on PSHB back in Durban, Botanic Gardens. So I heard you were talking about traps. How have the traps been working? Are they being able to trap the number of the beetles? Like, are they decreasing? Or because in Durban, back in Botanical Gardens, the tree were surviving after the infestation. So I just wanted to know that.
Yeah, I was in Durban Botanical Gardens myself in 2020, and I got some good samples there. I must say, I agree with you. The trees were surviving quite well. I didn't notice, absolute devastation there. It looked healthy overall. The traps that we use at the moment are called funnel traps. So it'll be an hourglass shape with a lure at the top. The lure is typically something like quisiverol. We're not even 100% sure why it works. It is something with the pheromones. Hopefully, if we unravel the genome sequence, we can understand that a bit better. But we have a lure. It attracts them to the trap. They fall into the bottom funnel, which we will fill with a thick liquid like propylene glycol, which preserves them quite nicely as well. And these traps work fairly well. As I said, in Vergelegen, 50 traps caught 5,000 beetles. Whether that's enough to draw them all out of the trees into traps, I doubt it, but it's at least enough to do surveys, to get samples for scientific study and that kind of thing.
Oh, great. Thanks, Luyanda. Thanks, Anandi. I just have a kind of a comment and a question myself, if I may. So just firstly, a comment. I think it's linked to really what Karen was saying. I mean, I also can't bear the thought of herbicides and chemicals and things. And I was thinking that I'm almost so tempted when we look at our massive problem of invasive alien trees. I'm tempted to hope that these beetles could be a kind of biocontrol for our poplar and our oaks that are escaping and invading our river courses and stuff. Yeah. But then it is a bit frightening, as you say, that it is in Ceralias, and so we don't know to what extent it could affect our native trees as well. So it's kind of tempting to think this could be our lifeline for these alien trees, but then at the same time, probably not, because it's just too dangerous. Like, it's something we don't really understand, and could really become a problem in our native trees as well. So my question for you then is just what advice would you give to, for example, cities where people like trees, they like the shade, and there's a place for that in our cities, which are very, very transformed. I'm not talking on national parks and things, but where we want trees. What do you suggest then? Like in Joburg, for example, where my in-laws stay, many of the trees have been ridden off. The community was going around and marking the trees with red bands to say which ones were infested. They've been falling over in the storms because they're totally they've died. And so the question is, what next? Do you have any suggestions for these cities and their planning?
Yeah, absolutely. So some cities are quite on top of things. The city of Cape Town's got a whole mandate on how to manage the shot hole borer, when to remove a tree, how to remove it, how to solarize it. And by removing heavily infested trees in the winter, when all the beetles are inside nestled, and they're not prone to escape because it's too cold, they're really lethargic when it's cold. You chop off that tree, you remove quite a large population of the guys, and you can get rid of that in a safe manner. That's one way to just try and bring numbers down. And then plant, as you said, we have a lot of the very popular garden trees are actually not supposed to be here. So I'm not so sad to see them go. So plant indigenous. And the first thing people will tell you is, oh, but the poplars grow fast, or they just make so much more shade. Just go buy a fat tree book and look at all the amazing indigenous trees we do have. And yes, the borer does attack indigenous trees as well, but they have a much better survival rate at the moment. And if we can get borer numbers down by removing their preferred hosts at times when the beetles are in there, and we're replacing it with stronger trees better suited to our environment that probably won't be so stressed because this is where they grow, that's at least in my mind a more sustainable strategy. I'm never saying, you know, completely getting rid of the problem, but reducing things to potentially a more manageable and a lower level. And if people are looking for what kind of indigenous trees to plant, ping me, ping your local botanical garden, or there are a lot of horticulturists around with amazing knowledge. All of the mature tree nurseries will be able to help.
Oh, thank you, Anandi. That's really great advice. And I mean, in Joburg, it's also, there are just so many amazing trees and the people here, really, there's a lot of people who are very passionate about it. So I'm sure they could find some good trees.
And I think, sorry,
no, go for it.
The sad part though, is that, you know, 50 years of growth on a tree is the impressive tree, and that will be lost. So, you know, it's not without its losses. Yes.
Yeah, no, absolutely. Thanks, Anandi. I was thinking, I saw Luyanda raised her hand. I don't know if you've got another question. I was thinking to make that the last and then hand over to the chair for his last words and his question about what on earth do we do if they arrive at Kirstenbosch. Luyanda, did you have a question? Did you want to ask a last one?
It was more for comment on what you were saying, because in my research I saw that, because I was looking on the more exotic side on what will happen to those trees, and when I did my research I found out that Durban Botanic Gardens has removed all of the exotic and started planting more indigenous trees, in which like decreased the number of infestation levels and erythrinas, one of those trees that were doing so well. So I think, going more indigenous is preferably would be better.
Yeah.
Thanks, Luyanda. Yeah, local is lekker. Awesome. Okay, so I think I'm going to hand over to our chair now just to close and to thank you officially. But from my side, Anandi, thank you. That was really wonderful. And I really loved how you handled all the questions. We really enjoyed learning from you.
Thanks for having me, guys. It was a lot of fun.
Awesome. Chair, can I hand over to you? Thank you.
And thanks, Anandi. That was a
fantastic your video talk. And we want to see your face.
And thanks, Anandi. That was a fantastic talk. I really enjoyed it, especially looking at the parallels with COVID. And yeah, I was hoping you'd give them nice enigmatic names like Omicron and stuff rather than PH4 and your things. But it's really cool seeing the research and all the different aspects, all the different branches coming together into one coherent, nice picture. So that's fantastic. And yeah, I hope you'll be able to help save some of the trees. And yeah, so that we can get on top of this. And of course, the fact that you're in Pietermaritzburg and everything's made easier by the fact that we've got the Zooming session. And yeah, things have changed for us. And I guess over the next 5-10 years, our cities are going to change as well with the types of trees and the different species and the way they look. So yeah, exciting times all around. And thanks for giving us a fantastic insight into it and helping us to explore it from an avenue that we don't even think about normally. Yes, the whole genomics and stuff. It's really cool fun. Really appreciate it. Thank you very much.
Thanks so much. I really appreciate the time.
Thank you.
Awesome. And Anandi, on behalf of Friends of Tokai Park, we'll send a little something in the post to you, a little gift voucher that's virtual, because we can't give you a bottle of wine or anything right now. But yeah. So
okay, next time we're both in Stellenbosch, we'll have some wine.
Yeah, that'll be awesome. Yeah. Okay, thank you so much. Oh, you'll have to join. Thank you so much, everybody. It was a lovely evening together. I hope you all enjoyed it as much as I did. Thank you for staying with us for our AGM. And yeah, I wish you a wonderful rest of your week and weekend. Yeah, be safe, everybody. Feel free to unmute and turn your videos on as you say goodbye. It's been nice to enjoy this with you. Bye-bye. Thanks, Becca.
Thanks, everyone. Bye.
Thanks, Anandi. Bye-bye. So good to see all the faces.
Everybody, thank you very much.
Thanks, Mike. Thanks, Karen. Nice to see you, Jenny. Bye, everyone. Becca, such a great smile. Hi, Leila. Bye, Linda. Cheers, everybody.
Bye, Alana.
Bye.
Bye-bye. Hi, Noskamanda.
Bye.
Leander, good to see your face. Awesome. Cheers, everybody. Have a lovely night.
Thank you for a great presentation and meeting. Good night.
Thank you,
Thanks
Ian,
for
for
the
your
questions,
interaction. Bye-bye. And thanks, Babalwa, for joining us. So good to have Sandbox here. Hi, perfect. There's one of our herpetologists. Yay. Awesome. Bye, everybody. Bye, Jay.
Bye.